This is the Discovery Files podcast from the U.S. National Science Foundation. I'm Nate Podker. Most living creatures, from colorful insects to charismatic panda bears, reveal themselves visually and are routinely photographed by humans from all walks of life. These photos represent a massive amount of image data that is inspiring a new scientific field called Imageomics, in which machine learning tools are being used by biologists to analyze the animals these photographs capture. We're joined by Dr. Tanya Berger-Wolf, a professor of computer science engineering, electrical and computer engineering, and evolution, ecology, and organismal biology at The Ohio State University, where in October 2021, she established the NSF Harnessing the Data Revolution Imageomics Institute. Professor Berger-Wolf, thank you so much for joining us today.
Thank you for having me here.
So I get to talk to a lot of people that discover cool stuff, but never anybody that's founded a new field of study. So what is Imageomics?
So it's the omics of images. Imageomics, right? This is the field of discovering biological information from images, particularly focusing on traits and phenotypes. So traits are the things that we consider important about animals, we can observe, and we can connect to function, often through genotype. So things that started with Mendel's discovery of the size, color, shape of the peas, and the pea plant, and the pea pod, and the pea flower. And that's how we started on the pathway to genetics and then genomics. But traits can be at the individual level, so the things like color, shape, size, weight, height, the pattern of the zebra stripes. They can be at the population level, the variation of the colors and shapes, but they can also vary during an individual lifetimes. Things like behavior or pregnant, sick, healthy, juvenile, adult have different proportions. The things like the sort of plants, so not only animals have traits, certainly plants as well, like peas, but the width and length of the leaf as it grows, the ratios of all this as the nutrition and the soil changes and this amount of sunlight changes, all of these are traits. And they're the product of genes, environment, and the combinations of the two. Now, the traits are not just important to observe and understand, but it is important to connect them to function and the context and the understanding of why. Not only what we're seeing, but why is it changing? Why do animals have traits? What is the intra-species trait variability within the species, and how is it related to the variability in the habitat or the conditions of that species along a particular gradient? And so, as humans, we're very biased by what we look at, what we can see, and what we consider important in terms of traits and everything else for that matter. There's so many things that we're not even capable of seeing because our apparatus that we're using to see is highly unreliable and is limited. We, for example, do not see we don't have enough acuity to distinguish red-orange variability in polymorphic moth because that did not evolve for our benefit. That variability evolved for birds, right? To fool the birds and to signal something to their own species, insects. Our vision is very, very different. There are also traits that are not even in the visual spectrum at all. There's chemical, the acoustic, and all the other kinds of sensory channels that we're not great at perceiving. And so we're missing a whole bunch of the world. And that translates into missing understanding of a whole bunch of the world. So where technology comes in, it makes the invisible visible. Because we're not only can bring in images from the same spectrum that we humans are seeing, but from a whole other range of spectra. And we can make sense of that. We can extract information from that and then connect it to a whole other wealth of data that then really not only finds new traits, connects them to different contexts, and starts through these patterns of connections understand the function of these traits and how they evolve, why they evolve, and what is their function in the also environmental context, in the ecological context, and understanding the truly maybe the life on Earth.
Right. There's a lot of other areas that it can spread into and applications. So thinking about the way we're visually analyzing things, seeing spectrum, is the image-based approach in machine learning why that's the best strategy? Because you can take in so much data that we can't process just as an average person.
Well, there's several reasons why we start with images. First of all, with all the technologies that is sensing the natural world, and think remote sensing, autonomous vehicles underground, underwater, on the ground, and in the air, on-body sensors, in stationary sensors such as motion-activated camera, digital cameras, camera traps, acoustic sensors that also, by the way, the spectral images are images, as well as these devices that we all are using and take copious amounts of images. In fact, if you think back, how many of us have taken a picture of a living organism? Probably everybody in the last week. Because, by the way, humans are living organisms. And every time we take a picture of something, there's probably a living organism that we're not noticing, whether it's an insect, a plant, or something else. And it's not only images are by far the most abundant and readily available source of information about everything today, but it is also the technology for processing information is the most advanced in two modalities, not surprisingly, images, because we are visual animals, and language, right? So all the large language models today, because we're also very verbal animals. That's what makes us so special and unique. But leveraging both that wealth of data and the technology that has advanced in those modalities really is a good place to start for us if we want to test the notion, can technology and computing bring something new to science?
And I think the answer is yes, unequivocally. But thinking about the machine learning tools, I guess, I want to break it out just a little bit more. How you're using it to bring out biological traits. So first, people to connect the dots, how are you using machine learning to bring out biological traits from photos?
So, when you start with observable traits, simple things like the color, shape, size, location, we want to, the first step, we want to go beyond saying this is the species of bird, but say this is the species of bird because it has yellow belly, black behind the beak, and it wobbles when it walks, and it makes this kind of call and lives in that kind of habitat. All of those are traits, right? And understanding that it's not only nice to have, it does two things. One, it pushes machine learning, right? And it pushes us in a good direction of interpretable AI, interpretable machine learning, to how do we provide evidence for an answer, this is this kind of bird. But that also allows us to quantify that trait variability. What is the variation? We can look at the entire species across all its range, for example, and that yellow belly, is it the same shade of yellow, right? Or does it change? And if it does, why? Is it the same length of beak? That's where Darwin started his study of finches and understanding the foundation of variability within species and within a family of birds, right, in this case, and why that length of beak changes in different habitats and how it changes with different diets. And that was the foundation of evolution, right, evolutionary theory. So understanding variability of traits, observable traits, we can today measure the length of the beak from photographs, has led us already to the foundation of evolutionary theory and understanding the evolution of life on Earth. Where else can it be that if we add all the traits that we have missed, right? Of haven't been able to quantify. There are traits that we can observe. For example, the zebra stripes and leopard spots. But we cannot quantify the similarity between two zebra stripe patterns, because we're very good as humans at quantifying similarity of faces. We have evolved to recognize faces. We have a whole infrastructure.
It's kind of our language.
Yes, we're so visual and we're so focused on the face, right? That there's a whole infrastructure in our brain that allows us to recognize faces and see faces even when there are non-faces. If you look at the clouds long enough or any pattern of dots, after about 3 minutes, you're going to start seeing faces of something. But and we are very good at saying, oh, the baby looks like its mom, the baby looks like its dad, even in lions, not just humans. Or it looks like that celebrity, right? But we are really, really bad. If I give you 2 sets of zebra pictures, and I say, are those 2 more similar to each other than these 2? We cannot do that. We cannot tell. No amount of training will allow us to be able to do it, which means we cannot answer the question whether stripe patterns in zebras or spots in leopards or spots on eggs are heritable, right? Are they inherited from mother to and father to child? Right? Is there a genetic signal in those patterns? And if so, what is the mechanism for heritability and what is the function? Are birds using the spots on the eggs to recognize kin, right? To recognize eggs from their own nest and those that are related versus unrelated eggs that are trying to, trick the mother to raise as her own? And the answer is yes, they do use the pattern, the visual, but we can't right now quantify this. And computational approaches for the first time let us do this.
I want to ask you about some of the sourcing, because one of the interesting things that has come up in some of the work you're doing is how you might let the average person in on this by taking advantage of the pictures just anybody takes of anywhere. So what are some of the ways you're getting images to this end from the field?
Yes, this is one of the great things that comes out of developing methods of using images as the primary source of information about life, is that it doesn't have to be carefully selected scientific images. It can be any images. As I said, all these, social media postings of a cute squirrel or birds. There are many, many, many pictures of birds or flowers or industrious spiders. They're now big source of scientific information. They're now source of biodiversity data because we can process them. And there's by far more data out there on social media about biodiversity that is completely untapped until now. And all it takes for somebody is just take a picture, to participate in the scientific process and conservation, right? To contribute to this. And not only that, but we are very intentional about developing technology that brings people in. So, for example, we developed a whole platform based on the ability to recognize individual animals from photographs. So this is not just species, but down to using those zebra stripes, using those zebra stripes and leopard spots and the unique shape of a whale's fluke and the dorsal fin of a dolphin, we can now say not only zebra, giraffe, whale, or turtle, but Zippy the zebra, Joe the giraffe, and Terry the turtle, and Willie the whale. And so now we can use images together with information on when and where it was taken to track and count animals. And we can open up the counting and tracking, right, to the whole world and to everybody. And so this platform that we built based on this technology is Wild Book, and it's part of the nonprofit Wild Me and Conservation X Labs. That is, everybody can contribute, but also built an intelligent agent that we're now all very familiar with. But this one is very much, much, much simpler than the types of agents that we're hearing about in the last few years. it really just scours social media and finds posts that are relevant to species that we can identify individually. And importantly, not only it brings in that data, but the first thing that we do, the agent tells the poster, hey, at 2 minutes, 46 seconds in your video, there's the whale shark that we can recognize. And here's all the other information that we know about this
animal. Here's where we saw it 100 miles from here.
And everybody else saw over the last 10 years. And people respond. The most common response we get, wow, this is amazing. How can I help? Right? That how can I help part, that engages them because they didn't realize that they could contribute by just taking a picture, by just taking a video on their vacation, and they typically have more than that, just one video.
Right. That's just the one they thought was good enough to post.
Exactly. They probably have hundreds. Right. And not only of that whale shark, of everything else, something that smeared or not maybe charismatic enough. And then they typically contribute then all of it. And we now can have wealth of that data bringing in, right? Yes, it's still biased, but so is every other source of data that we use, whether it's camera traps or scientific projects focused on one particular tiny habitat, right?
Still going to be some bias to it. So one of the things you've talked about in your lectures with this is kind of the potential use and how it might help conservation efforts. And one of the points you bring up a lot is data deficiency. So can you talk a little bit about, I don't think people are aware of this, that we don't know about these things?
Yes. People have often shocked to discover that, first of all, there is estimated only about 10 million, with an M, species of plants, fungi, and animals on Earth. It's a shockingly small number. Most people think that they're in the billions, or at least 100 millions. There's only about 10 million. And that's including all the millions of species of beetles. By far, probably the most prolific part. And about 1 million or more are threatened with extinction. So that's more than 10% of the world's biodiversity. But even more shocking thing is that we're losing species faster than we can name them. There is about 2 million total named species of non-bacterial species. And the International Union for Conservation of Nature Red List, so this is the official organization, international organization that tracks the biodiversity of the world. So this is also the organization when we say that the species are endangered, it's because the Species Commission of IUCN Red List, by certain metrics, quantitative metrics, determined that the status of that species is endangered. And so of the 160,000 species about that they're tracking, very biased towards vertebrates, with tiny fraction of those beetles being even noted. About 20,000, more than 20,000 of those, the official conservation status is data deficient, meaning that we don't have any information for even the basic metrics. How many, where they are, where they're going, is the population trend up, down, stable? Nothing. And about 60,000 more, the population trend is unknown. And these are not obscure species. Killer whales, orcas, the biggest whale, the biggest dolphin in the ocean is data deficient. I mean, come on. You can track that species, and yet we don't have enough data. Polar bears, the iconic conservation species, population trend is unknown. If we don't know enough about those kinds of species, what hope is there for beetles? Right? The workhorses of the ecosystems. And so if you're keeping track, that's more than half. Data deficient to population trend is unknown. More than half the species of those 160,000 that IUCN Red List is tracking to begin with. There's a massive data problem. There's a massive data problem because the bulk of the data that we have is also about charismatic species, about species that are common, species that are in the middle of their ecosystem, species. We know a lot about North America and Europe, but that is not where the world's biodiversity is highest. And it's not also where we're losing biodiversity at the highest rate, right? Which is the tropics, which is the South America and Sub-Saharan Africa. And so we don't have data where we're losing it fastest. We will not know what we've lost, right? And you can't protect what you don't know. You can't recover it. We don't have data at the edges of our ecosystems. We don't have data on rare, cryptic species that are hard to observe, inaccessible ecosystems. We don't have data on ecosystems that are changing fastest, that are subject to the highest pressure of biodiversity loss drivers. So that's the data gap, right? And it's vast. And while I'm a computer scientist, I am not going to go out in the field and put in policies and do the hard work that it takes to mitigate the loss of biodiversity, to stop and reverse it in some cases. Data problem is certainly something that computer scientists can do something about.
All right. So not to end it on a downer, I want to talk a little bit about how you've now started an institute about this field. And I want to talk to you about what your goals are with the institute and what the next steps are. How are you going to bring this out into the world and put it into effect to where you're impacting this stuff?
So we started in the Imageomics Institute, and that is to mobilize all the image data and connect it truly to the science of the biology, biodiversity, ecology. But we also NSF funded the AI and Biodiversity Change Global Climate Center that brings together ecologists, biodiversity scientists, and computer scientists to explicitly, not just from images, but from all modalities, help fill that data gap and derive new ways of understanding the biodiversity and ecosystem function at a both very, very local scale, where it changes most, where it's subjected to climate pressure most, where it's most vulnerable, and also where we have the least data and understanding. So to drive new ways to analyze the data and that result in new biodiversity science. Hopefully bringing in not only the fantastic team that is at the core of AI and Biodiversity Change Global Climate Center, that is also with high intersection with the Imageomics Institute, but also engaging the broader machine learning community, the broader computer science community, because they're hard computational problems there, and the broader biological community in this endeavor, and then translating it into deployed solutions that can move the conservation management, that can move the biodiversity management and practice, and support the policies that actually make a difference. Because if we do this right, then we can truly, I believe deeply that we can meet that challenge. We can truly not only provide the deep scientific understanding, but also translate it into action that makes a difference in the field.
Maybe change the world. Special thanks to Tanya Berger-Wolf. For the Discovery Files, I'm Nate Potker. Discover how the U.S. National Science Foundation is advancing research at nsf.gov.